Oh wow. Sorry, it's blurry.
Hmm.
Hi, Shane.
How are you?
Good, how are you?
Good, thank you.
Okay, so I think honestly I was just like distracted doing cars.
Hmm.
I felt weird about like the test yesterday. And second social I like rushed through and I finished early. But overall, like I don't know. I got the highest second social score I've ever gotten.
Hmm.
And it took like 40 minutes to take it.
Yeah. Which test was this?
This was two.
Okay, and what's the breakdown?
I have 124 Chem Phys,
Mhm.
127 Cars, 125 Biochem, and 128 Second Soc.
Got it. Okay.
Yeah, I don't know. So like a little bit different, but I feel like CP went up. Wasn't it 123 last time?
Mhm. You said
So
for cars you felt distracted.
yeah, honestly, it just like felt off.
Hmm.
And I like zoned out at some point and then I was rushing at the end. Like I don't really know what was going on. I don't like anticipate that happening again though. I think it was just I started later than I was supposed to because I had ended up having a meeting at 11 on Sunday. So I had to like go to the meeting, come home. So I only started after.
Okay.
But yeah. Anyway, should we review?
Hmm.
Jack Weston or Norm?
Hmm. You could hide, yeah.
But then I'm not gonna know which ones I got wrong.
So, so yeah, let's try this. So you could hit like the Chem Phys section, and in the filter, you can have it filter for like wrong or flagged. Yep, you could do.
Incorrect and flagged? What if I got it correct and it was flagged?
Yeah, so let's do this. Go back to the correctness all has a flag. Yeah, and just hit that purple filter button.
There are things that I got wrong.
So hit that purple filter button. Yeah. All right, now I'm gonna just write down which questions these are. So number 2, number 4, number 6, number 17, 25, 28, 36, 43, 56. So it seems like one of them that you got right that you flagged. Uh,
Okay.
now you
28.
can go, yeah. Now you can filter it for the incorrect. Yep. Purple filter button. Yep. And now review all with Jack Weston, top right, and hide solutions.
Okay.
Okay. So why don't you walk me through your thought process for this?
I was looking at AMC and I remember there was another question
Hmm?
about this that I was really confused by. So this is the AMC structure.
Mhm.
I remember, was this the tetrapeptide? I picked four, I think, because I thought it would attach from like a carbon. And like one is attaching from like I'm assuming the hydrogen would leave
Well,
and it
they're
would form
they're telling
like
us that they're
ammo
they're telling us that they're using something
over.
in the passage. So there's probably no way to answer this without looking at the passage information. Mhm.
Okay, let's just read it. That a high porphyrin compound was capable of inhibiting the activity of proteasomes. Proteasome inhibitors exhibit anti-tumor activity in cell culture, inducing apoptosis by disrupting the regular degradation of pro-growth cell cycle proteins. Consequently, researchers were interested in determining whether this activity could be improved by chemical modification of porphyrin, figure 1. Then we have general structure features of porphyrin compounds studied. The effect of porphyrins on the chymotrypsin-like activity of purified rabbit proteasome was measured by by following the increase of fluorescence as 7-amino 4-methylcoumarin AMC was liberated from the peptide succinyl LLVY AAMC. This is amino acid structure, right? As a result of peptide bond hydrolysis. Chymotrypsin is a digestive enzyme that catalyzes the hydrolysis of peptide bonds that follow an aromatic residue. In the absence of proteasome activity, no increase in fluorescence was observed. What is the site to the model tetrapeptide? This?
Mm-hmm.
So it's attached to the LLVY. Y is STY. It's polar.
So how is this attached to AMC?
A peptide bond.
Yep.
At what site though? Here.
Uh-huh. So you want to definitely like, especially
Read carefully.
in the studies, right?
Yeah.
So that indicates that there's something important in the passage that we need to answer this question.
Mm-hmm. Okay.
I think you just kind of randomly chose something, but yep, you see it's a peptide bond. And that is, do you remember what a functional group a peptide bond was?
It's in wait, a peptide bond? I thought that it was like a linkage between a uh hydroxyl and a
Mm,
COA carboxylic.
so what do you think it is? A peptide bond?
Maybe a hydroxyl group if it's talking about hydrolysis.
Well, what does hydrolysis mean?
Moving in OH group.
No.
Hydrolysis, like adding.
So, okay, so hydrolysis, you definitely need to know this. So hydrolysis is breaking bonds through the addition of water. What's the opposite of that?
Ligate.
Nope.
Hydrolysis breaks down. Like through the addition of water. I know what it is. I don't remember the name. It's not ligating.
So just think about the opposite of hydrolysis. So what does hydro mean?
Water.
So hydrolysis,
Adding water.
so hydrolysis involves adding water or removing water?
Removing it.
Well,
Well, through the removal of water, it removes an OH and an H.
why
Okay.
would
Yeah.
it be hydro then if we're removing a water?
Didn't you just say that hyd that it is removing a water?
Mm, mm, no, I'm saying what if so what yeah, I'm saying what is hydrolysis? So if hydro means water, we would need to be adding water, right?
Does it add water to break down the thing?
Mhm.
Okay, so then the opposite would be removing the water.
Yep. So what can that be called? If you haven't had any water all day,
Dehydration.
uh-huh. And then the second part, what's
Dehydration reaction or dehydration synthesis.
what's the opposite of lysis?
Synthesis.
Hmm.
Okay.
So if you're forming a bond, in this case, a peptide bond, you are, or the molecules are undergoing dehydration synthesis, also known as condensation.
Okay, so the same.
And then
Condensation.
opposite would be breaking a bond by adding water. That's hydrolysis.
Okay.
You definitely want to know that. Now, in terms of the functional group for a peptide bond, what would that be?
I have no idea.
So a amide, this bond, because what you have happening in an amino acid is, so I guess like next time, so we're gonna go over a lot of stuff today. So next time we meet though, I think it's a good idea for me to for the first let's say 5 or 10 minutes, just kind of quiz you on some of this stuff, just to make sure that you have it down. So for instance, I'm going to draw, and drawing things out always helps. Like that's something that I also kind of realized recently, like about not necessarily you, but a lot of my students don't like to write things down. So, or draw things. But I recall when I was studying for this test and taking this test that I would always be writing stuff down. And I think
Mhm.
that helped jog your memory. Like when I had you 2 days ago, draw you know, the action potential, like depolarization thing. Like you were able to get the right shape. It was just shifted up above. So yeah, I would say if you're having a hard time remembering something, maybe start writing or drawing something out that can help jog your memory. So for instance, I'm going to draw a regular amino acid, like a nondescript amino acid. So And that's something you should be able to draw. So basically, I'm having this amine group. And also you can kind of think of the word amino acid to see which is in the front. The front part will be the amine group. And then it'll be bound to some carbon that'll be bound to some R group,
So
which, what's up?
that part doesn't matter, or it's carboxylic acid?
Oh, sorry, what was that?
Is it carboxylic acid for the second one? Or it doesn't matter what the arc?
Oh, so yeah, the R group is just going to be whatever side group exists for whichever amino acid, because each amino acid, they'll have the same structure that I'm about to draw for you, but they'll differ in their R groups, right?
Mm-hmm.
So I'm just writing a nondescript amino acid with R group just written as an R group, but that R group would change depending on whichever amino acid you're looking at. But yeah, you're going to have a carboxylic acid after that. Um, so I'll just draw it like this, like without it being in the Zwitter ion form.
Mm-hmm.
And then I'll draw the next one, but I'll draw it like slightly different, like this. And then let's say this is R1, this is R2, and that, like that, right? So these are two separate amino acids that will, if we want them to combine, to synthesize, because synthesize means you're bringing things together to build something. Lysis is breaking or splitting something. So what I'm going to show you here is that this, what I put in this light blue box, happens to look like water, right? OH and an H. So that's exactly what happens. The OH from this carboxyl.
Forms the high H2O, which is hydrolysis.
Yeah, and so the stuff in this light blue box will now disappear. And a bond will form between this nitrogen here and then this like carbonyl carbon here. So this will look like pretty much like, I'll start off drawing the same thing. And the new bond that formed is this orange bond,
Mm-hmm.
right? And that is your amide, aka peptide bond.
Okay.
And I always say that for things like, peptide bond between, amino acids, for sugars, you'll have your glycosidic bonds. For fats, you'll have. So for all those types of bonds, you want to know, like, sure, it's a peptide bond, but you also want to know what functional group it is in terms of organic chemistry. Um, and it's for the peptide bond, it's an amide bond. And
Can you go over like
Mhm,
which ones go for which, please? Like amino acids are peptide bonds.
mhm.
Um, what other kinds of bonds are there?
Yep, I can go over those. So quickly though, this peptide bond has double bond characteristics, meaning, the nitrogen has two extra, like an extra lone pair.
mm-hmm.
It has five total and one bond here, one bond here, one bond here, three bonds. So there's still five. I'm sorry, there's still three valence electrons left. So these can go here, forming a double bond, and this can go up there, giving you the O minus, and then the double bonded NH for this.
Mm-hmm.
Like I didn't draw the rest of the stuff. R1, blah, blah, blah. But this just indicates to you that this amide bond, peptide bond, is quite strong and
Okay.
difficult to break. So, but if you were to break it, you would break it through the addition of water. And so
How
the
would the water come off? Like,
sorry, that's just NH2 over there. I didn't draw the entire thing. I was just trying to draw what happens at this peptide bond region. But yeah, if you add water to break it, it'll be hydrolysis. But if you remove water to make the bond, it'll be dehydration
hydration synthesis.
synthesis, also condensation.
Condensation synthesis or just condensation?
Just condensation.
Okay.
But the dehydration synthesis is more like
Like,
uh, just literally the opposite of the hydrolysis, like hydro adding, dehydro removing water,
mhm.
lysis breaking, synthesis making. So that as well. So
Yeah.
that's what you would get for this. And you're asking something,
I wanted to go over like what types of bonds go for which.
were you asking something about the hydrolysis also though?
No, I don't think so.
Okay. Because you know, I would say that a hydrolase would be a type of enzyme class that would do the hydrolysis part.
Mm-hmm.
Um, and but okay, yeah. So peptide bonds form between amino acids. So now the other things we can have are, for instance, so for like a triglyceride, you'll have this molecule, which is glycerol,
Mm-hmm.
and you'll have, and it's basically just like 3 carbons bound to 3 hydroxyls, right?
Yes.
And then we can have our fatty acid, which has a carboxylic acid functional group, and then a long aliphatic tail or a long hydrophobic, like hydrocarbon tail. It's kind of like maybe like Saturday when we went over that question that was like some compound one, I think it was like, with like the disulfide or the cysteines and blah blah blah, and then there's this hydrophobic part in the first part, which is just a long hydrocarbon chain. So yeah, so we have our fatty acid here. And what happens here is, well, dehydration synthesis, which means that we're breaking this time like this bond, right? In the light blue box.
Yeah. And that adds an H2O, so it's also hydrolysis.
You mean dehydration synthesis? Because we're making a bond between the two.
Oh, so it's removing.
Mhm.
But then what's different between that and this?
Well, I mean, they're both.
Both dehydration.
Yeah, you're gonna see that for all
Okay.
all these. So,
All
uh,
right.
So yeah, so now what functional group does this is this one? Is this over here?
Mm, that's carboxylic acid.
Nope. It was here. So let's do this. Let's do yeah.
Amide? What
What?
did I do?
Oh, you just click the different tab, I guess.
No, I didn't. It's the same tab. I hate that it shows.
Yep. Hit the score report. Chem Phys. Filter by incorrect. Yep. Hi. Yeah. Okay. So where we're okay, so yeah, this over here. Oh, sorry. No, I'm gonna have you do this on the left. So here, I'll write it above so you can see it better. So let's start with this. What's this?
Aldehyde,
Very good. And that,
ketone,
and that,
carboxylic acid,
and that,
ester,
and that.
amide.
Well, yeah, you got it down. So now look at this.
But it doesn't have a negative charge, so it's not carboxylic acid.
Correct.
I don't know what that is.
So what's the other one that hasn't, an O there?
Ester?
Yep. So the R means just like a bunch of carbons.
Okay.
Yeah. So yeah, you have your ester here.
Okay.
Have you ever seen like omega-3 uh supplements and stuff?
Probably, yeah.
So um have you ever seen them like write it out as like esters?
I don't know. Honestly, is that omega-3 like a vitamin or
Kind of,
pro?
kind of. It's a fatty acid that has a lot of really good uh
Oh, is it the thing that's in fish?
oh, like fish oil.
Yeah.
Mhm.
Okay.
Yep. So okay, so this is a fat that we've just made here.
Yes.
And so the bonds in this triglyceride
Mm.
are ester bonds.
Okay.
The bonds within a fat are esters.
And it makes a fatty acid chain.
Yeah. Now the glycosidic one's a bit different. They will look like, let's say, this. Oops. Like that. This is a
Wait, where did you draw
on the far left over here.
that? Okay.
You see him?
Yeah.
Yep. This is a hemiacetal Okay.
Okay.
And, it's like this X thing, but it's kind of hard to see in a actual molecule, but what it would really look like would be like, let's say, this is a glucose ring with an OH here. So what exists, I'm going to use, let's see, purple. So this is the R, this is the H, this is the OH, this is,
Mhm.
what's this one?
Oh.
Uh-huh. And so R, because the O is attached to more carbons.
Oh, so it's an ester again?
Uh, so no, something else. This one.
But
Right.
you're asking about this specifically, this branch of it, not the whole thing. It's a hemiacid. Um,
This entire thing is the hemiacetal, but
yeah.
even if I was talking about this one, that wouldn't be an ester because you would need a carbonyl group.
Okay.
Um, so but yes, this is a hemiacetal, okay? Now
Uh-huh.
that's what's going to be at the end of a sugar. And if it's, hemi, it could be oxidized, which means that it can be a reducing sugar, right?
Hmm.
But if this forms a bond with another sugar, like let's say a 1,6 bond,
Wouldn't
what's up? Yeah, I didn't write out all the.
I know, but wouldn't it be, I'm trying to, wouldn't it be a dehydration synthesis because you would be taking these two off?
Yep. And so now we're left with, we still have that OR, we still have this R, we still have this H. Sorry, it's hard to see. Let me try doing this.
Mhm.
So
You like select the image and move it up or no?
if Zoom doesn't let me do that if you're sharing the screen, which is stupid.
Uh,
But um, so yeah, OR here, R here, H here.
oh, I can move it.
Uh-huh. Yep. So yeah, and then so now we don't have the OH anymore. We have R, H, OR, and another OR.
No, it's
So now
yes.
instead of it being a hemiacetal, which is like a half of an acetal,
It's a full acetal.
full-fledged acetal, yeah. Now, the hemi, can be a reducing sugar,
Hmm.
but the acetal, so and the hemi is what's on the end of um a sugar like glucose here. Uh, but when you form a bond, a glycosidic bond, it becomes an acetal or a ketal, but it no longer will be just hemi, which means it's no longer capable of reducing things.
Okay.
So we have your amide bonds, which are the peptide bonds. We have your ester bonds, which are the bonds within a fat, like a lipid. And we have your acetals, which are the glycosidic bonds, the bonds between sugars. And they're all formed through dehydration synthesis, also known as condensation. And they all could be broken again through hydrolysis. And it's not like you just like add water and it breaks. like it kind of looks like that with hydrolysis. It looks like you just add water and it breaks, but you're
Mhm.
gonna need some type of, you know, first of all, some type of enzyme. Like,
Substrate.
so like for instance, a protease is an enzyme that breaks proteins, breaks those peptide bonds. So that's something that can cause hydrolysis of the amide peptide bond. You could have a lipase that breaks the bonds of a fat. Um, and you could have a, what is it? A, but I forgot the name, but you'll also have an enzyme that breaks glycosidic bonds. And they'll all be broken through hydrolysis. And so, yeah, those are the different types. I mean, there's also, of course, um, the bonds of like the phosphate bonds, like in ATP, for instance, like this,
Uh-huh.
etc. And so here we have, I'll use pink here. So we have our, well, here we have our like phosphodiester bond because we have, well, the phospho part.
But where's the OR?
Uh,
Is it
oh, like that.
this?
Uh, just what's connected to the oxygen here could be the R.
Okay.
But but yeah, this over here would be phosphodiester because we have an OR here. We kind of have an OR there. I mean, it's an O connected to another phosphorus atom, but it'll still be, yeah, it'll be an ester, but a phosphoester, which just means that instead of that R group being a carbon, it's just a phosphorus.
Okay.
Yes. So that would be your phospho
Diester bond.
bond. Yeah. Here is a image in the chat. You can display that. There you go. So we see that on the left, we have some, ribose sugar with, a phosphate and stuff, and then another one below it. And yeah, I know what I wrote is, yeah,
Oh, whoa.
that's cool.
I do not know how to do this. Okay, never mind.
Yeah, you can just move. Yeah. So yeah, here we go. So we see that. That's why also like for DNA and stuff, you need like a 3' end because that's where you have your OH group. Now, I'm pretty sure that, well, so okay, so basically we have our nucleotide here and here, two separate ones, and we want to connect them together. And we connect them together by getting rid of a H2O again.
Mm-hmm.
So what do you see happening here?
You said that the 3' end is where it attaches, so maybe this hydrogen comes off and this OH comes off.
Uh-huh. And so you get
The
your
oxygen bonds, right?
phosphodiester bond, right? Because you have the phosphorus here, and then you have the OR here,
Uh-huh.
R here.
Uh-huh.
And so that would be the bonds between the phosphate backbones of some type of like, DNA, RNA, or even like some type of, ADP kind of thing.
Mm-hmm.
So,
Okay.
yep. So yeah, you can take a screenshot of all this so you kind of have it. But yeah, key thing here is forming bonds through dehydration synthesis, which means H2O leaves, and that's what the little light blue rectangles are that I made. And then you'll get the bond, which is specific to whatever thing you're working with. And if you wanted to break that bond, you would need to do the opposite of dehydration synthesis, which is hydrolysis.
Okay.
Yep. So,
The only energy equation that I remember is E equals HF.
so let's think about this conceptually first.
Okay. Light energy, I know that there's a constant, which is this, right?
Planck's constant.
Yeah.
Okay. Now, remember how I told you fluorescence is super high yield?
Mm-hmm.
So, what do you remember about fluorescence?
Energy of a photon,
Okay.
right?
Mhm. So let's look at what they said in the passage about it. Yeah. Continue.
The general experiment procedure was as following. One purified rabbit proteasome was incubated in the presence of porphyrin at 37 Celsius for 30 minutes. The reaction was initiated by the addition of peptide and intensity of fluorescence emission at 440 nanometers, excitation at 360 nanometers was monitored for 20 minutes.
Okay. So they're giving us some information about fluorescence. What information are they giving us?
The intensity and excitation.
Uh, but what values are they giving us here?
Nanometers, which is wavelength.
All right. And yeah, what's the difference between like why do we have two different ones?
Um, because when it was excited from this to this one, or
Mhm.
maybe even more, but this is where it was fluorescing.
Tell me, sorry, say that again.
This is the like wavelength that there was fluorescence at, like not at this one.
So what does that mean? What does that mean?
So the wavelength was, well, not the wavelength, but the like it was excited. Um, like it's like a charge. I don't know if charge is the right word, but it was raised from like one wavelength to another. That means that I'm pretty sure that means that the photons were like stimulated.
So you're thinking,
Kind of like electrons, right? Like when they're like they get excited and they move faster.
so they can get excited, which means that they will be they will move to a higher energy shell.
Photons or electrons? That's electrons.
Yeah, so if you think about um the photon, if the photon has a certain at least a bait like a minimum amount of energy that's needed,
Mm-hmm.
uh let's say that's between the two um that's between the two shells, right? So like something like what could be something like what are you like maybe like visualizing in your head, or like what would be something that maybe you can draw to help jog your memory?
For this one, I don't know because I don't know. I'm still so like rough with like physics has gotten a lot better, but this I like don't know.
Okay. Is your phone away?
Yeah.
Okay. So uh okay, so yeah, let's talk about this. So let's say that we have three like shells where electrons can be.
Mm-hmm.
Let's say the energy difference between n is equal to 1 and n is equal to 2 is 1 electron volt. And let's say between 2 and 3 is 0.5 electron volts, which means the
The
total,
jump from 1 to 2 is harder than the jump from 2 to 3, right?
yeah. So like, yeah, the electrons will start at their ground state, which is n is equal to 1.
Mm-hmm.
Now, if we had our, whoops, 1, 2, 3, and we have our electron in the one shell, and we shot a photon with energy of 0.99 electron volts, what happens?
It would be closer to 2.
So
It didn't cross that 1 EV, so it's still N equals 1.
good. So it won't be closer to 2.
Okay. Yeah. So it's on the first shell.
What if we had shot two photons with this much energy?
Wouldn't it still be on 1?
Uh-huh. Why?
Why?
Yeah.
Because they're each their own like photon. It doesn't combine.
They can't just this why can't this just go up to 0.99 and then this goes up to like
1.8. Because they're separate. They can't just add and combine.
good. Good. Now, what if you increase the intensity?
The intensities brings this up.
When you say bring this up?
Like brings the EV like electron volt.
So
Like
think about what is intensity.
intensity is like the way that I'm thinking about it in terms of light, it's like let's say a light bulb, like if you I don't know if charged it, but like if you added more watts to it and it like is brighter, then it's more intense light, right?
Huh? Yep. Now, would increasing the intensity help the situation here?
Yes, because you would help it go up to n equals 2.
So what is what is important about intensity, because they will love to ask this type of question. Intensity is not
Equal to,
energy,
okay.
right? So they will love, they will definitely test you on this, and also they will give choices that'll be like, okay, like, because clearly we don't have enough energy in these photons to be absorbed, right?
But intensity, that's why it's not like the equation doesn't have EV in it because it's different, right?
Oh, what are you talking about?
Like when I was trying to explain what like I thought intensity to be, I said watts, not electron volts.
Well, what is so an electron volt is supposed to represent uh what quantity?
Energy,
Correct. And watts, what about that?
power,
Which is
energy over time.
Mhm. So like rate at which energy is released or produced or whatever. Now, intensity has units of watts per square meter. So that's why, like, so I probably asked you this before, but like in the summer, I guess it's still technically summer. Are we closer to the sun or further from the sun?
Closer.
We're further.
What? But the sun is like,
So now let me ask you this. Let's say this is you, and this is the sun at, I don't know, 9 a.m. And this is the sun at noon. What time do you think it'll feel the hottest? When it's at noon?
mm-hmm.
All right. Now let's think about why, right?
Because it's like when it's directly shining down.
Uh-huh. Yes. So when it's at an angle like this, the light rays,
Does that have to do with like the axis that like the planet is at towards the sun, not the distance from it?
yeah. So for instance, here, the sun's rays are spread apart over this larger distance compared to, I'll use green. If when it's directly above you, the light's rays are spread out over a smaller area, right? And if
Mhm.
intensity is inversely proportional to the area, you could increase intensity by reducing the area. So that's why it's hotter at noon than it is at a different time. You're still getting, the sun's rays, right? Sun's photons, but you're spreading it over like if you spread it over a smaller area, it's going to give you a higher intensity. So
Okay.
same thing with the sun, you got it. If our northern hemisphere is pointed towards the sun's rays will hit the northern hemisphere at less of an like oblique angle and kind of more directly, as opposed to if the Earth was tilted away, meaning the northern hemisphere being tilted away, then the sun's rays will be spread apart more because it's hitting it at like an oblique angle.
Okay.
So like um so yes, so intensity, its unit is power per area, and it's not the same thing as energy. That make sure you get, because
Mhm.
they'll like to test you on that by kind of trying to trick you into thinking that they're the same thing. Now intensity is essentially proportional to the number of, at least for light, the number of photons, which, as you said, is proportional to brightness.
Mm-hmm.
So if you increase the intensity of this 0.99 electron volt light, you're really just increasing the number of those photons. And we know that if we have 2 of them, it's not going to make a difference. So if we have 2 trillion of them, still not going to make a difference. Still doesn't have enough energy. All right?
Mm-hmm.
Good. Now, let's shoot one with 1 electron volt. Now, what can happen?
Now it goes 10 equals 2.
Very good. So what happens is, think about it like this. It's like the atom catches this high energy photon, and it's like, oh, what do I do with this? Gives it to an electron in the ground state, and the ground state electron's like, all right, I'll bring it up to the n is equal to 2. So the electron to the photon gets absorbed by the atom, and that means a ground state electron gets excited to the n is equal to 2 shell here.
Mm-hmm.
And if it's at a higher energy level, it's inherently less stable. So it will want to then relax back down to the n is equal to 1 shell and give away that energy by emitting a photon with 1 electron volt.
So as soon as it rises up, it wants to get rid of it?
Yeah.
What's the point of it going up then?
Well, we can see, like, you'll see, like, right now. So now let's say we have a photon. I'm gonna make it blue with 1.5 electron volts. Now what happens?
1.5, it rises 10 equals 2. Oh, no, it goes all the way 10 equals 3.
Good.
And if there's something there, it wants to get rid of 1.5, right?
Now, so up until this point, I'm just kind of showing you like for a regular atom. However, if the molecule contains a lot of conjugation, aka resonance, aka electron delocalization, aka pi bonding, you'll see all of those terms being used to describe the same thing.
Mm-hmm.
So, and you could even, if you ever look up like, like melanin.
Like, we talked about it, like skin cancer. Yeah.
So yeah, melanin, like, and dyes and pigments, they'll have a lot of conjugation. And figure 2, we see that the fluorescent tag is what they're showing us, and there's a lot of conjugation here, right?
Right.
Conjugation, just think of, like, for instance, this, which means this can go here to give you this, aka resonance.
Okay.
And resonance does what to a molecule?
Stabilizes it.
Exactly. So what can happen is, although this 1.5 electron volt photon is absorbed, if we had this as a dye or a pigment or something with, a lot of conjugation, it can relax that electron down from n is equal to 3 to n is equal to 2 without having to emit a photon yet. Just due to the fact that it can undergo resonance stabilization, can relax that electron from 3 to 2.
I don't get it. How?
So it's like, So let's say the same example with like, you catch a photon and it's like, all right, you give it to an electron and the electron has to go up to the excited state at n is equal to 3. But let's say the electron is like, I'm feeling very anxious. I'm going to do some meditation, some breathing exercises, aka resonance stabilization, and it'll allow that electron to relax a little bit without having to emit a photon, just through the fact that it has resonance stabilization can reduce that energy level a bit.
Why would it want to do that?
So let's see, right? So now, so let's at least agree that the electron that got excited to n is equal to 3 by absorbing that photon can get relaxed to n is equal to 2 without emitting a photon.
Okay.
Does that make sense? Okay. Now, when it does eventually go from 2 back to 1, it will emit a photon. However, what would the energy be of the photon that it emits?
Mm, I don't know.
So where is the electron going? Like from where to where?
Going down.
Uh-huh. So from which shell to which shell? So
Right.
yep, so that's what it does. I put like a dotted kind of arrow there to indicate that's what it can do through resonance.
Oh, so then the rest of it goes down like from 2 to 1.
Yep. And
Oh,
that has an energy level of,
1. The electron volt.
yep. So it will emit a photon with 1 electron volt, right? So what is this showing us? This is showing us that in order for fluorescence to occur, the energy of the photon that is absorbed is larger than the energy of the photon emitted. So I'll kind of write that in red. So basically, energy of the,
Um,
what's up?
a photon is equal to energy emitted.
That would be, sorry. Uh, so okay. So the energy of the photon absorbed will be larger than the energy of the photon emitted,
Okay.
right? Because the photon absorbed here is 1.5 electron volts, and the photon emitted is 1 electron volt, right? So the energy of the photon absorbed is larger than the energy of the photon emitted. Now, what about wavelength of the photon absorbed compared to wavelength of the photon emitted?
I don't know. Wouldn't they also be the same?
Well, did we say anything is the same so far for fluorescence?
No, it can't be because wavelength is associated with intensity.
No. So what? So okay, you said that you remembered the energy of a photon like equation. So what was that?
E of photon?
The energy of a photon? Uh-huh.
E photon. I remembered energy equation was E equals HF, and then I remembered like E photon was in a different equation, and I don't remember that one.
What if you didn't have F, for instance?
Isn't it lambda over?
So you could write it out like in your notebook.
like to use my notebook when we're meeting because I get distracted by what I'm writing
Mhm.
down. I don't pay attention to what you're saying. Okay. Okay.
Mhm.
Okay. Equals H. If F's not there, it's I don't remember.
So if you don't remember the formula, the units will help you,
Frequency in hertz.
which is also 1/seconds.
Is it lambda over time?
So let's see, right? So
But we don't have time here. 1/seconds. Oh, 30 minutes.
no. So don't worry about any of that stuff right now. So so you said energy of a photon is HF, right?
Mhm.
Now we want to figure out how we can do this if we don't have a frequency term.
Right.
So if the frequency term is in 1/seconds, right?
Mm-hmm.
We can figure out a way to solve for that with two other values.
I know that wavelength is one of them.
All right, what unit would that be?
Lambda.
Unit?
Oh, wavelength. Nanometers.
We could just consider it to be meters. Now, what can you do with these if you look at the units?
Meters per second.
Yep, if you do what?
If you divide them.
Uh, try that.
Wait. Time, multiply.
Okay, because if you multiplied lambda by f, you would get, well,
Mm.
meters times 1/seconds, which is meters per second, right?
Mhm. So speed.
So it's what?
Speed.
All right, now what can we plug in for speed?
I don't know, acceleration.
Well, why would you plug in acceleration for speed? They're different, right?
Yeah.
So what can we plug in for speed? What's a photon?
Like the light particle.
So what do you think the speed would be for it?
The speed of light.
Yep.
So 6.
No, don't focus on that right now. So because we want to So you need to be able to tell like the relationship between energy and wavelength.
Mm-hmm.
So if we say that lambda, I'll make it in red. So if we're saying that lambda times frequency is the velocity, and if it's light, it's equal to c, right?
Mm-hmm.
So now, if you go back,
I thought C was like from the I over C equation,
the what?
like current, voltage, I over C, current over.
So little c is the speed of light in a vacuum, right?
Okay.
You definitely have to know that. So speed of light in a vacuum, right? So now back here, energy of a photon, if we didn't have frequency, what can we write?
C over lambda
Uh-huh.
H.
Because over here, if you solve for frequency, you divide lambda on both sides, you get frequency is equal to c over lambda, right?
Yeah.
So energy of a photon is equal to hf. But if you don't have f, you'll always have c because that's a constant. And if they're giving you wavelength, you could just use wavelength. But what's the proportionality between the energy of a photon and the wavelength?
Inversely.
Correct. So back here or down here,
Mm-hmm.
so we know for fluorescence that the energy of the photon absorbed is greater than the energy of the photon emitted, right?
Mm-hmm.
Now tell me what that means for the wavelength of the photon absorbed and the wavelength of the photon emitted.
That means that the wavelength emitted will be greater than the wavelength absorbed.
Exactly. Right? So if you go back to the passage where they talk about fluorescence, what are they giving us?
The intensity and the excitation.
Well, sorry, so
Um,
they're telling us that something, okay, so the excitation would be this or this.
absorbed.
So they're saying that it gets excited at 360 nanometers and that it gets emitted at 440 nanometers.
But it's not energy, it's intensity. We have a whole thing.
Well, no. That's so that's saying that that's what they're showing us in this figure,
Okay.
right? But something I think something I said to you like a really long time ago was anytime they give you the wavelength of light, you know you have everything you need to figure out its energy as well as its frequency. Because energy would just be this, right?
Yeah.
H are constants. So if you know the wavelength, you can figure out the energy.
So since it's the denominator, which wavelength?
And also this shows that if you only know the wavelength, you could still find the frequency.
Okay. Can I ask a question?
Yep.
Say, so we know HC. I'm gonna go out on a limb and assume that C is the Wait, no, we know it. H is 3 e to the 8th, right? Or is it the other way around? That's the speed of light.
So what? So what did we write for the speed? It would have to be if you multiply which terms?
Speed is lambda times frequency.
Yeah, wavelength times frequency, right? And so that'll be C. And so, here they're telling us that the fluorescence emissions at 440 and the excitations at 360, which is exactly what we see, what we want to see for fluorescence, because fluorescence is all about emitted photon has less energy than absorbed photon.
Right.
So that's fluorescence. So now, if we look at this question here, they're asking which expression gives the amount of light energy in joules per photon that is converted to other forms between the fluorescence excitation and emission events. So step one, what is it asking? Always start that way with questions.
Amount of light energy converted to other forms. What does that mean, converted to other forms? Between the excitation and emission events. Is that the difference between, like, the energy lost?
So it's pretty much asking what's happening there.
Huh.
Which is what you were saying.
So I subtract 440 and 360 nanometers.
Uh-huh. Because going up here is what happens when you have that 360 nanometers absorbed. And here is when you have the 440 nanometers emitted.
Mhm.
So this must be the energy that is converted into other forms.
Oh. And is this the value that I plug in for lambda? So like 440 minus 360 is 80?
Where are you showing me?
I'm not, I'm just asking. So if we're looking for the difference between 360 and 440, it's 80.
So,
That would be the lambda value that we plug in here.
mhm,
And then HC are constants, and that's the numerator. So that looks like this one.
mhm. Yep, because if you do energy of the photon absorbed, it will be hc over the 360 nanometers times, -9 minus the energy of the photon at 440, so hc over 440 e -9. And yep, we could just factor out the hc to get 1 over, so hc times 1 over.
Was never -6. What am I? Micro is 6 and nano is 9.
Sorry, what are you saying? What are you asking?
Like I thought that nano was e to the -6. That's 9.
No. Yeah. It's, what's up?
Because milli's 3. I thought nano then micro. Is it micro then nano?
Micro then nano, yeah.
Okay, so micro is 6, nano 9. Okay.
Yeah, like, AKA, like, milli is a thousandth. Well, centi is 100th. No, deci is a 10th. Centi is 100th.
Milli.
Milli is a 1,000th, and then it starts going by threes. So, micro is a millionth. Nano is a billionth.
Pico.
Pico is a trillionth.
Okay, and that's 12.
Yep, and then there's
I never went past pico.
femto, -15, ado, negative 18, and other stuff that you don't have to know. But yeah, so here we go. So first of all, that's how you do it all. So why don't you take a screenshot, because you got to know this stuff about fluorescence. It's super high yield, and you will undoubtedly see it. But let's say that we didn't know what to do. Like, I always want to supply my students with multiple things that can be done if you don't know, right? And so they're asking for a type of energy, right?
Mhm.
So this is just HC,
Yes.
right? And that isn't going to give us energy.
Yeah, neither is B.
B is HC times lambda.
That
That's
won't.
that's not going to give us energy either.
The one that could give the answer is D, but this is emitted or not emitted.
Yep, no, it is emitted, yep.
Light.
Yep. So D could possibly, like, actually is a value of energy, because you do have HC over lambda, and C has it as well. So at the very least, you should be able to eliminate A and B.
Mhm.
But if we're talking about fluorescence, we're talking about energy, being lost through the state of the resonance stabilization. So, and they're just saying other forms, and that's pretty much what they're implying.
Okay,
Yeah.
cool. It's the total number. Okay, so I was thinking of like what we went over with the like two-dimensional thing would be SP2, right? And then anything with like
So
tertiary.
when you say
I mean secondary structure, like I'm thinking of.
oh, that would be for like proteins and stuff.
But
But
I thought that was what the thing was here, that anything with like D orbital was already like tertiary.
uh, so it's not gonna
We
be
went over like there was something that you were like, this is like, I don't like this topic.
oh, you're thinking of bond geometries.
Yeah, maybe.
That can get annoying once you have the d orbitals and stuff.
So
But
this is different.
uh no, this is still about bond geometry and stuff, and hybridized orbitals, but it's just asking. So let's do like a little refresher there. So sp2 sp3. Now, do you remember what types of bond geometry would, or bond angles, or
I thought it was like single, double, tertiary.
oh, so tertiary, I guess what you're saying, what you mean to say is triple bond,
Okay, all right, yeah.
right? Now,
Structure and like the proteins, like secondary, tertiary.
yep, that could be something with proteins, secondary structure, tertiary structure, but it can also be something with, like, this is a secondary alcohol, primary alcohol, tertiary alcohol, so it could be that as well.
Number of carbons that it's bound.
Yep. As well as, if it loses the OH group, it'll be a secondary carbocation, etc. So those are the contexts where secondary primary can occur. But yeah, for this, we want to know that. So think about it like this: sp predominantly triple bonds, sp2 predominantly double bonds, sp3 predominantly single bonds.
Okay.
And so if you think about something with a triple bond, like
It would be X.
HCN, it is a linear structure, which means that the bond angle is 180 degrees. Right?
Uh-huh.
sp2 will just have a double bond, so it can look like this. And this is,
240.
so this is this, or not that part, this over here. So imagine 360 is the entire thing, but if we're breaking it apart into 3 segments, they will be 120 degrees.
Oh, it gets smaller. Okay.
Yeah, so this is linear up here. This is trigonal planar. It's flat.
Okay.
And then sp3 single bonds, that's just going to be like, for instance, your methane, which is a tetrahedral, which means a bond angle of 109.5. Okay.
Uh-huh.
So that's what those mean. Now let's try the question.
sp2 hybridized carbon atoms present in the fluorophore.
So we're looking for what?
Number of carbon atoms that are double bonded.
That have what type of?
sp2
Yep, with this shape, right?
Yeah.
All right, go for it.
I think that no, I think it's this one, the fluorescent AAMC fluorophore. Okay. One, two, three, 4, 5.
What are you counting?
Carbon double bonds.
Uh-huh. Double bond consists of, what are they asking us to count?
How many carbon atoms
Yep.
are sp2 hybridized?
Yep.
So that means that for everyone that I counted, there's actually 2, so it's 10.
Almost.
Okay, like this double bond is 1, 2
Keep going.
carbons.
Yep, keep going.
So 2, 4, 6, 8, 9. Because this is an oxygen.
Yeah.
Okay, so 9.
Yeah. But you want,
Because this isn't double bonded, so this is a carbon atom, but it's in this linear.
no, wait, no. So, yeah, wait.
Oh, tetrahedral for CH3.
So these are all, as you said, sp2, right?
Well, we were looking for the ones with a double bond, but this one is a single bond.
No, we're looking for the sp2 carbon atoms.
Yes, I know.
So let's,
I know. So the answer is 9. I was just like clarifying that like CH3 would have a different
so
config.
1, 2, 3, 4, 5, 6, 7, 8, 9. Now the CH3 here, like if I could just redraw this, and it looks like this, right?
Yeah.
It's still 120 degrees, meaning it's still,
As that's cut off of this carbon. I'm saying that the bond between C and the 3 hydrogens is not. Like not looking at this carbon, only this carbon that's in the CH3. It's not sp2.
so uh so since it's a part of this, it'll still be that,
So
because
isn't that another one?
uh not really, just because, like, if we had
It might as well be the R group, is that what?
Yeah, because if we had a ketone here, right, it doesn't matter like if this is like, for instance, compare this to that. Like this is still part of this, so it'll still be sp2 hybridized,
Okay.
right?
All right.
But main thing here is you want to be very, very careful about the precision in the language, because when you did it fast, you were gonna say you basically said 10 of them, right?
Because I counted this as a double bond, but this isn't a carbon bonded to a carbon, so there's only 9.
Yeah, because you first just counted the number of double bonds. So 1, 2, 3, 4, 5, and then I think you doubled it. But they're asking for the number of sp2 carbon atoms.
Right.
And that's where you see that for this double bond, there's an oxygen and a carbon rather than 2 carbons. So you want to definitely be very, very careful about that and make sure that you're reading every, word that the question is asking you. Okay. Right.
Okay.
Yep.
I don't know what a lactase is, but I
Oh,
was
I didn't even see this. All right, let's see.
it's
Well, if it ends in ASE, it must be what?
enzyme.
Yep.
But is it lactase or lactase? I've never heard of CC.
Uh, I guess it's lactase, but of course that's what the passage is for. So you want to take a look at that.
Oh, activity. Okay.
Like, I would
I
answer
I
this.
know I read it when I was doing it yesterday, but like I literally like the passages leave my mind instantaneously.
Yep, yep, yep. That's fine.
The structure of okay, should we just read it? Lactases are copper-containing metalloenzymes found in the wood rotting fungi. They catalyze the exergonic decomposition of lignin, a major structural component of wood. The active site of lactase consists of 4 copper ions arranged in the shape of a kite. The ions are classified into 3 groups, T1, 2, and 3, based on their chemical and spectroscopic characteristics. So 1, 2, 3. And then, wait. These are the same. Okay, these are the same configurations.
Okay. So we get to see the structure of the active site of this thing.
Yeah.
And we could answer this question based on that. So what do you see?
I see sulfur.
What can that indicate?
There's only 2 um amino acids that have sulfur, and that's cysteine and
Hmm?
methionine.
Yeah. And
It's not SH, and methionine's not here. I picked cysteine.
so wait. So go back to that figure. Tell me where you see these things.
In T1.
So you see 2 different types, right?
Yeah.
So what can that tell you?
Isn't this what you were saying with the hemialdehyde or
Nope. We're dealing with amino acids here.
I know, I was just looking at
So the hemi stuff is with like acidic bonds, but so okay, so we have something here and something here, right?
uh-huh.
Are these the same?
No.
All right, how are they different?
the bonds, sulfur's bonds. So
Uh-huh.
this sulfur has 3. This one only has 2.
So you could think about it like this. So this amino acid has a methyl group attached to the sulfur. Because if something looked like this, would you be able to tell what like the lines mean? Because I know you were having some trouble with that before. Like the line notation.
Like I feel like I've learned so much. Like it could be that it's linear, but amino acids. You're talking about methyl or ethyl.
No,
Like
kinda, yeah. Like
it's a primary.
so I guess what I'm asking is just like let's say that we have I'm gonna use a different example. Let's say that we have this.
That's a primary alcohol.
Uh-huh. Now, do you know like what is there? Like,
No, but that's why we draw it as like a squiggly line.
oh, sorry. Let's just say that this is straight up how it looks like.
Okay. It's
No squiggly line. Now, uh, what is
carbon.
is it just carbon or
Well, it depends. This carbon only has 1 bond, so that means that there's 3 hydrogens.
good? Yeah. So this is a CH3, right?
Yeah.
And what about this guy?
Um, H2.
Uh-huh. Just
H.
CH2, right? And then the OH. So yeah, I was just trying to make sure that you were.
Oh, okay. Yeah, no, that's I knew.
So for this, you have a CH3 here, right?
Yes.
A methyl group essentially. And that can maybe help if you think about methionine, like the meth part, having that methyl group. So
Mhm.
that's this, your methionine. Okay. Now, this, what do you think this one would be?
Sulfur, CH2.
Uh-huh. So we don't have a methyl attached to that sulfur, right?
Yeah. So that one's cysteine.
Yeah. And so a cysteine looks like this. It has what's known as a thiol group.
Okay, that's what I said. I like just since methane and methane isn't like in the answers, and this is SH.
Well, let's not pay attention to that right now. So let's just focus on this. So the thiol group, right? Like what's a thiol group? So the thi part indicates sulfur, and the OL part indicates alcohol. So imagine, so like if it was just this, it would be a an alcohol, right?
Mm-hmm.
You see what I drew over here?
Yeah.
But if we replace the O of the alcohol with an H, I'm sorry, with a sulfur, it'll be a thiol. Okay. And the thing with thiols is that they can form disulfide bonds with one another.
Okay.
So cysteines look like this with the thiol groups, and they can form disulfide bonds. Methionines are like this with a methyl group attached to the sulfur, and these cannot form disulfide bonds.
Should I be looking for other amino acids since they're talking about 4 copper ions?
I mean, yeah, but I just want to make
Okay.
sure that you know that if you see this, that you know that
Iodine and this is cysteine.
this is cysteine, and then also that cysteine has thiol groups, which means that it can participate in disulfide bonds. That's really important.
Okay.
So, okay. Now you can look for other amino acids because they're asking which one has the greatest abundance.
These two have the same configure. Well, there's two T3s, so I think I need to be looking in this area. I see ring structures. Isn't that phenylalanine has a ring and then another like ring on top of it. But it doesn't look like this.
Okay.
Right? So is it tyrosine? I can't remember. I thought it was threonine that was next to it. Tyrosine was different.
What about threonine?
I don't remember the structure. Oh,
So let's go back to what we talked about regarding kinases.
okay.
So what are kinases?
No.
Uh, try to like think. We went over it a lot of times. It's a very high yield thing. Okay, so kinases are transferases that transfer phosphate groups, and they use three amino acids.
STY.
Yeah. Now, what do those three have in common?
The OH group.
Yes. And if you see an OH group, what do you want to think?
Most common nucleophile.
Good. And most common electrophile would be something with a carbonyl group, but could also be a phosphate group because a phosphate group is pretty similar to a carbonyl group in that it has a bunch of bonds to electronegative oxygen, right? Now, oh, and then STY, like what's the S called? Like, what's the amino acid for that the S? Serine, and then the T.
Threonine.
And then the Y.
Phenylalanine.
So
No, ST, they're both T's. Threonine, it's not tyrosine.
tyrosine.
Then weren't there three that had T's and there was one that was non-polar?
I don't know what you mean when you say T's, but
Like three amino acids that start with the letter T.
oh, I mean, yeah, I get maybe, yeah, sure. But the important thing is to recognize that STY all have hydroxyl groups. And in this active site, do we see any hydroxyl groups?
No,
So what can we eliminate?
tyrosine T.
Okay.
And yeah,
Now, what can we do?
we have a lot of amides.
Do
Or
we?
no, we don't, because we don't have any oxygen. Never mind. We have a lot of amines. So the amines are
So we can see that clearly that, this is the most abundant, right? Like it's everywhere.
yeah,
So we could eliminate another choice here.
histidine.
Um,
I don't know the structure. Phenylalanine I know has the like ring and then another ring on top of it, and I'm not seeing that here.
so no, I'm saying that we agree that this is the most common or most abundant.
Oh, so then cysteine we can cross out, but I already crossed out cysteine in my head because I knew that there was a lot of these ones.
Okay.
So I'm trying to figure out between phenylalanine and histidine.
Okay. Now the phen part indicates a benzene ring.
Yeah. Which this isn't a benzene ring. It's a pentene.
Okay.
So it's not phenylalanine, which makes it histidine.
Yes.
Okay.
Yeah, and this is called an imidazole ring. The one in the histidine. Imidazole ring. It's called that because, well, it's a heterocyclic ring, meaning that it contains not just carbons, but nitrogens as well.
Okay.
So let's think about how we answer this, right? So you
I
look,
found sulfurs. So I listed out the two that I knew that had sulfur in them, which is cysteine and methionine.
okay,
And then we knew that those were both present here, but then we looked at like what there was a lot of
mhm,
in this compound,
mhm,
and that was like these
mhm.
imidazole.
Yep. So we knew that cysteine was present, but that's not the most abundant one. So we can get rid of cysteine.
And then
Yeah. Um,
we I just how did we get to the kinase?
no,
And
the kinase I mentioned to help because you were kind of confused with STY, right?
okay.
Like you were like I think you thought that like I don't know something about STY that wasn't right. So I mentioned the kinase to indicate to you that they use STY, which they use to phosphorylate things, and that means STY must have OH groups. And that's when I asked, do you see any OH groups, or anything like that? And you said no, and that meant that we could eliminate tyrosine because I think you were saying that tyrosine is
Okay, yeah, I understand.
yeah. So that STY thing with kinase is super important and high yield. So I'm adding it to I'm keeping like a list of like things to ask you, I guess, next time we meet to make sure you have it down. But yeah, since we didn't see any hydroxyl groups, we can get rid of tyrosine. Then you're left with phenylalanine and histidine. And if you remember that the phene part is a benzene, like that's what it stands for. And we don't see any benzene rings here, which means that it can't be the phenylalanine,
Mm-hmm. Okay.
which by the way is just this. Whereas the tyrosine is this with an OH also.
That's the problem, is that phenylalanine and tyrosine are right next to each other, or like when you're learning them, and they both have the ring structures. So I have to remember that like phenylalanine has the double, like it has another thing like over here.
No, it's what I drew. So that's why the STY is important because the STY shows you that we're looking at hydroxyl groups. And so if we have a tyrosine, we're going to have sure, we're going to have a phenyl group, but more importantly, we're going to have the hydroxyl group. Whereas phenylalanine, we just have the phenyl, the benzene ring.
Okay.
Yeah. You're thinking of tryptophan. Tryptophan has 2 rings,
Okay.
right? Uh, it has a
Benzene ring and then a pentene on top of it
yep,
that has an H in it.
yep, yeah.
Okay. All right. Oh wait, let me pick.
Okay.
Compound 3 catalyzed reaction. I was really confused because
So let's make sure we understand what it's asking.
what structure is formed when this is a substrate of a reaction catalyzed by lactase, which means that the substrate and the lactase have to bind. And we know that the active site consists of 4 copper ions. That's what I'm like, I can't understand where it would bond and what it would attach to and where it would lose. Like
Well,
this,
so it's asking, so tell me again what it's asking.
it's asking for the structure, like what would be the product basically when compound 3 reacts with lactase, because they
Okay.
have to bond because substrate enzyme.
Now, I'm just wondering why you're looking at this part, like figure 1.
Because this is lactase.
Yeah, but does figure 1 tell you anything about like the product formed when lactase is doing its thing? No.
No, no.
So we want to look for where it tells us like what lactase actually does, right?
Catalyzes the oxidation of phenols.
There you go. Oh, and then so what could a phenol be?
It's alcohol
Oh,
of
well,
fen.
and phene part means
I don't know.
we just did it. Like in that previous question with the phenylalanine. What does the phenylalanine look like?
The benzene
So the
ring.
fen part means benzene.
Okay, so it's a benzene attached to an alcohol.
Yep.
Okay. Reaction 1 is an example. But these aren't alcohols.
We're
Saying that reaction 1 is an example.
yeah.
How is this an alcohol? I thought that it had to have like
I mean, well, what would an alcohol have to have?
the like carbons.
I mean, there's
Um,
carbons there.
Yeah, but we've always drawn it as like a carbon tail.
Uh, so okay, let's think about what an alcohol means. Like, what would it mean to be an alcohol? Like functional group wise.
Hydroxyl group.
Mhm. Yep. Like ethanol is just this, right? So we have our fen part, which is the benzene, and the OL part, which are the hydroxyl groups, right?
Oh, all right, I understand.
Mhm.
Um, okay, so
So yeah, what's happening in reaction 1?
It's uh the wait a second. Oh, so both of these hydrogens are coming off, and then the oxygen is getting resonance stabilized. But okay, there's two of them. Where does the second oxygen go?
So what type of reaction is this?
This is it's from hydrolysis, oxidation. Oh, well,
And
I knew it's oxidation. I'm just trying to like go back to like dehydration or hydro.
well, those are not oxidation though.
All right, never mind.
And if it was hydrolysis, you would have water as a reactant, right? But oxidation is what's happening here. And you want to know how functional groups change when you do oxidation and reduction.
Well,
So,
you know that when it's oxidized, it gains a negative charge.
uh, I'm talking about like for functional groups,
Okay, all right.
like if I had CH2 like this, and I oxidize it, what will it turn into?
CH minus.
Hmm. So there's in the PDF folder, like you don't have to look at it now, but there's I have a PDF that is just biochem, like general redox, like reactions of like what actually happens. And you're gonna have three things happen, like one of three. Either you're removing hydrogens, you're adding oxygens, or you're adding bonds to oxygens.
Okay.
So this alkane here will first lose hydrogens and produce a double bond like this, right? And then it will add a bond to oxygen, forming an alcohol. And then it will add bonds to the oxygen, giving you, some ketone or some aldehyde, and so on and so forth. Like then, so yeah, you want to know that because here you want to be able to see that an alcohol can be oxidized. In this case, this is a secondary alcohol, so it can get oxidized into a ketone. But that's what we're doing here. So again, when you read a question, make sure you like try to rephrase it so that you understand what it's asking, and then use that to figure out what to do. Like
Okay.
for this question, you went all the way up to like something on the left, like figure one, the active site. But they're asking about what structure is formed when compound 3 is the substrate of the lactase catalyzed reaction. So you want to go to, first of all, compound 3 is here, right? And lactase, what that does is mentioned here, right? So I think you're having trouble trying to find where in the passage things are related to. So yeah, so you want to know what it's asking. And from there, you can kind of figure out where in the passage to go.
Okay. Do you mind if I went to the bathroom for a second?
Yeah, yeah. Go for it.
Thanks. Okay.
Mhm.
All right. So would it break the nitrogen?
Well, why would it do that? Like, what does it say that it's doing?
When compound 3 is a substrate in this reaction.
Mm-hmm.
Would that make it like a reactant like here?
So,
It's
so what does the lacase do?
oxidize, it catalyzes the oxidation of phenols.
Yep.
Okay.
And then we have an example.
Yes.
And what's happening there?
The hydrogens are removed and it becomes a double bond.
So the hydroxyls are turning into carbonyls, right?
Yes.
All right. So now, instead of compound 1, just put in compound 3.
How is this a carbonyl? It's
Carbon.
it's not. Oh, yes, never mind. Yes. Wait.
A carbonyl is a carbon double bonded to an oxygen.
Oh, okay. Because I was like thinking of carboxylic acid, and I was like, this isn't carboxylic acid. But okay. All right. And then O2 becomes 2H2O. Because those are the two hydrogens that came off.
Okay.
And otherwise it had to have lost an oxygen because wouldn't it be 2O2?
Why?
Because where did the second oxygen go?
There's a 2 in front of this.
Mm.
But I think you're getting too caught up in the weeds here. So what does
Because
lacase do? Like, simplest way. Like, what does lacase do?
catalyzes.
What?
Catalysis.
Like, what do they catalyze?
Phenols. The oxidation of phenols. So this reaction, the OH2O.
Okay. So now imagine.
So so catalyze it like speeds up the reaction.
Yeah,
So the substrate
trying to figure out what.
binds the enzyme in order to do that. So compound 3 has to bind to the phenol that gets oxidized.
Okay.
So it's not C because this is the exact structure of compound 3.
Kind of, yeah.
Oh, the OH.
Okay.
Okay, wait. I get it. So it's not oxidizing the compound 3 because it's not like, because I started looking at like, okay, where did it break it? It started breaking like off of the benzene ring, which is where the nitrogens are. So then I was like, is it going to break the nitrogen and then do something there, which is what like A looks like?
No. So wait, hold on. So let's. So what does the lacase do?
It catalyzes the
Yeah.
oxidation of phenols.
Okay. And that's what it does.
Compound 3 is the substrate
So
in the reaction.
so what? How are you going from oxidation of phenols to breaking nitrogen bonds?
No, I'm saying that's not what it was doing. That was like what I
What is
okay, no. I was explaining like that's what I was thinking like the first time I did this problem. That's why I got it wrong. So I'm saying I don't want to do that this time.
okay? But let's talk about what to do like this time.
Okay. Um, I think that compound 1 has to bond with compound 3.
Right? So I don't know what you're doing here. So they're telling us that lacases catalyze the oxidation of phenols, and they give us an example. All right.
Right, but there's like the oxidation of phenols. So isn't this a phenol, but we can't touch it because it's the substrate. It's not the
The substrate is what the enzyme helps turn into a product.
okay. So these two are phenols.
Okay.
And then we have the hydroxyl groups
Well, if that's
turn into a double bond,
uh-huh.
which is D.
Yep.
So then why were they taking like half of it? Like what do A and B like accomplish?
I mean, those are just they're asking what happens if you take compound 3 and you add lacase. And we can see quite clearly that, well, they tell us it's oxidation of phenols, but if you don't know what an oxidation of phenol looks like, the example here is there to show you that we have hydroxyl groups turning into carbonyl groups.
All right, I understand. So like this is only an example. Like you shouldn't use this as a structure. We're only looking at compound 3.
Yeah, because it says literally it's an example, and the question is asking literally what happens when compound 3 is a substrate. Okay.
Okay.
So we see here that, so two things. We see here that lacases cause phenols to be oxidized. And if you didn't know what that looked like, which you should know, by the way, but if you didn't know what it looks like, you could just see that there's an example here. And the example shows what? It shows that you
Is
take
it then?
OH group and you turn it into a carbonyl group,
Okay.
right? And then they're asking what happens, go back. Then they're asking what happens when you use compound 3 instead, right? And if you go scroll down again on the left. And so we see compound 3 over here. And we know that from this example, lacase takes hydroxyls, turns them into carbonyls. So compound 3 take our hydroxyls, turn them into carbonyls. It's that simple.
Okay.
Because I think you were doing so you're like trying to add compound 1 to this for some reason, but you got to read the question. And
No, I read the question. It was like the example part that was throwing me off.
yeah, but why would you add compound 3 to compound 1, right? It says compound 3 is the substrate of the lacase, right? That means you compound 3 over here, you add lacase to it, and then it turns into what is what it's asking. We're not adding it to compound 1. So yeah.
So
Mhm.
I remember when we were going through, it's like there was a problem that was like when it's 1/2, it's the next like if 2 minutes is 1/2, then 4 minutes isn't the rest of the time, it's 1/4 of the time. So then I started thinking that it's
What
like
do you mean 1/4 of the time?
we were looking at a problem like
So what does a half-life mean?
it means like half the time it takes for half of the like thing to decay.
Good.
So that's why I crossed out A and B
Okay.
because half the time I remember we're doing a problem that was like if 2 minutes is the half-life of like what is 4 minutes, and I got it wrong because I said all of it will be gone, but it was 1/4 will be left.
Okay.
So right, I crossed out A and B. And then I said all the to decay into wait. This doesn't make sense. Half of it to decay into this. So D,
Mm-hmm.
because I didn't like understand what daughter nuclei were because I've heard of daughter cells, not daughter nuclei.
So now how do you know it's that and not?
Well, like reading over it, like half of all, why, how can it decay into itself? It doesn't make sense. It has to decay into something new.
Well, this isn't saying that it's decaying into itself. It
Well,
just
I picked C when I was going through it. Like I immediately crossed out A and B. I just didn't think that daughter nuclei was a thing, so I said daughter. Like,
sure. But this doesn't mean it's decaying into the same thing. It means that it's decaying into other things that happen to be radioactive.
okay,
But
I don't know why that's wrong.
well, because radio
It's
decay, half-life is decay of a radioactive, material, but it doesn't have to decay into other radioactive things.
okay. So that's like it's too extreme, is what you're saying. It's like a cars thing when
Uh,
it always has to, like you want to avoid that kind of a thing.
uh, it's just that what's always going to be true is that it decays into the daughter nuclei, kind of like the daughter whatever cells or gametes or whatever. But the daughter nuclei that it decays into may or may not be radioactive. Um, but C says that it has to be radioactive, and that's not always true. But D says a daughter nuclei, and that's always true because we always call the product the daughter nuclei. Yeah.
Okay.
Mhm.
Well, I don't know why it would increase the force to stand up.
Okay.
You'd want to decrease the force it takes you to stand up. Um,
What is this question asking?
like why do you need to like either lean your body forward or your legs like back
Mm-hmm.
in order to stand up
Mm-hmm.
to creating an angle kind of thing. Um, I was thinking of like force, like chair, person, and then standing up. Like you're creating like energy with your feet. I think I picked C because I was like when you swing your feet or like lean forward, you're like giving your like you're reducing the energy to stand up. But then I would just realize energy cannot be created or destroyed, so C is automatically wrong.
Not true. So you have a certain gravitational potential energy when you're sitting down,
Mhm.
and when you're standing up, you have a change in height, which means that you're actually increasing the energy.
Yeah, like MGH.
Mhm.
And if you lower the height,
Mm-hmm.
and you're lowering the gravitational potential energy. So it doesn't keep the body in equilibrium because there's still something changing.
So we want this guy to either put his feet below. If that happened, then
Body forward.
all of this is aligned. Alternatively, I'll use blue. If the person is leaning forward here, these are aligned as well. So,
Okay, let me think about this.
yep.
So normal force and gravitational force. But we crossed out A and that's the only one with force in the
Mm-hmm.
um it's not friction, is it?
Nope. Because like the friction could be like on the horizontal if maybe you slide your feet under the chair, but it's not going to be the thing that helps you get up, and it's not going to play a role in if you lean forward either.
Mm-hmm.
Have you ever done like squats, like barbell? Or
No, I know what you mean.
like because like let's say this is the barbell, and it's just going down like this, and you want it to be you know, like your butt to be like behind and your feet here, whoops, more like here to be aligned. Because let's say that it wasn't, right? Let's say that you did something like this with the barbell here, but I see people at the gym doing it like this all the time, which is why they're gonna mess up their knees. But if you just had it like this and like your butt's like here and they're not lined up, you're gonna just tip forward because you're creating a torque here. You're gonna topple forward like that. So like if you in this example here, um, if if he slides his feet underneath the chair, all of like the center of gravity of this person is going to be lined up, thereby not producing a torque. A torque would produce a rotational movement, which would cause the person to like tip over. But if you do that, or if you lean forward, the force vectors line up and can enable you to stand up without putting a torque or a rotational force on you, thereby keeping your body in equilibrium, particularly equilibrium dealing with the torque, like if this is like the clockwise torque and this can be like the counterclockwise torque, if they're equal and opposite, you're going to have that rotational equilibrium. So
Mm-hmm.
it's not going to be friction because that'll be like on the horizontal. It's not going to be energy because when we stand up, we're actually increasing our energy, at least in terms of gravitational potential energy, and it won't increase the force or change the force. But yeah, it's to line everything up so that we are in rotational equilibrium.
Okay.
Mhm. Like imagine this guy's feet like this, like
You're saying that it's like,
gonna fall backwards.
right. So it's keeping like,
Mhm.
okay.
In rotational equilibrium. Yep.
Circular dichroism spectroscopy measures the difference in absorbance of left and right circularly polarized light. This phenomenon arises when absorbers are in an asymmetric environment. Changes this environment, as when protein changes conformation, result in changes to the CD signal intensity. Asymmetry resulting from tertiary structural signals causes the largest increase in CD signal intensity. Secondary, tertiary. Good.
So, sorry, how are you able to answer that?
Asymmetry resulting from tertiary structural features causes the largest increase in CD signal intensity near the UV region
Oh,
of peptides.
uh, so, well, okay, like,
Like when I initially did it, I was thinking of like the charge of tryptophan and how would like
oh, so yeah, so there's something that they mentioned here that can indicate to us that, because I guess like you kind of like guess this being correct, but that's not entirely the reason why it's D. There's another thing. It's like right after the sentence that you just read.
the side chains of our aromatic amino acid residues absorb in this region.
Yep.
Side chain of tryptophan,
Yeah, aromatic.
like is a benzene ring.
So I mean, it doesn't have to be a benzene ring, but yeah, it'll be
It's
some type of ring. And since they're mentioning tryptophan, right, that is an aromatic ring, right?
Mhm.
So yeah, you're right about the tertiary part, but they could have said a different amino acid here. Like if they said one that wasn't aromatic, then our answer wouldn't be D. So I just want you to be aware of that.
Okay. Based on the relative energy of the absorbed electromagnetic radiation, which absorber a peptide bind or an aromatic ring exhibits an electronic excited state that is closer in energy to the ground state. Do I need electron? Well, neither of them. Oh, that's not what it's asking. Never mind. Um,
What is it asking?
which between a peptide bond or a aromatic side chain exhibit an electronic excited state closer to ground state energy based on relative energy of the absorbed electromagnetic radiation, which I think is like figure 1.
So
Like
what,
asymmetry causes the largest increase in intensity, which is different energies absorb in this region. Asymmetry does not impact, nor do the elements of secondary structure. So we did a problem that was like this, but I can't take.
so what is this question asking?
To see if peptide bonds are aromatic side chains. Um, when they absorb energy or at an excited state closer to the ground state. So lower excited state
Lower energy. Yep.
or energy for a peptide bond or an aromatic side chain.
No.
The wavelengths are the same. Absorbs, well, yeah.
Huh?
The far UV region versus the near UV region.
Okay. So the wavelengths are the same or they're not the same?
I just can't understand what the near UV, far UV region is. Does that mean that near UV is
Well, if
UV
you have,
light, which is closer to 290 than, oh, they're different. Wait, hold on.
yeah,
This has a lower wavelength,
which means
a lower energy.
write the formula.
For energy,
Yep.
E E equals HC over lambda.
Okay.
So the greater the wavelength, the lower the energy.
Yeah. So
So
remember,
that peptide bonds will have a lower energy.
yeah. So remember that that's inverse because you said higher. So you want to
Right.
write it down.
Okay.
So okay, yeah. So which ones?
So that means that we know it's not A or B. Absorbed photon energy is lower
Why?
because, well, we just calculated that E photon is lower for a peptide bond than for
How?
when I solved.
What do you
We
mean?
said that it was inversely proportional since peptide bonds have a higher wavelength than the
They have,
energy. Huh?
where do you see them saying that it's a higher wavelength?
Wait a second. The side chains of aromatic amino acids. Okay, so it's aromatic,
Mhm.
and these have a lower E photon.
Yeah. So you because you right away you just said that you would cross out A and B.
Yeah, I know.
So
It was a
higher,
stupid mistake. I like solved the
higher,
problem.
prior to that, I was asking if the wavelength goes up, the energy like goes where, and you said goes up. And then that's when I had you like think about the equation to see that they're inversely proportional. But you still like we're looking for the one of the smaller wavelengths. So maybe if you write it down, it can help. Like to write down like energy of a photon is inversely proportional to wavelength.
Okay.
So I would get into the habit of writing it out like this because if you don't, like you're gonna make mistakes like you were doing here.
All right. Okay. Three phosphanoinositide dependent protein kinase 1 phosphorylate specific serine or threonine residues in the conserved activation loops T loop of other serine threonine kinases, resulting in the activation of those proteins. Those other kinases possess a conserved PDK docking site known as the hydrophobic motif HM.
So this one you can just kind of look at this.
Mm-hmm.
That's the amino acid sequence.
What is F?
Um,
Phen Phenylalanine.
for the F, yep.
Okay. L is leucine.
Hmm.
G is gluten
Glycine.
glue. Huh?
Glycine.
Leucine.
I mean, sorry, the G is glycine.
Oh, glycine.
Hmm.
Phenylalanine,
Mm-hmm.
tyrosine, and tryptophan.
Nope.
Threonine.
Yeah, so you're still having trouble with STY.
So it's threonine and tryptophan that are in.
Nope. What is
Why? Tryptophan.
what does tryptophan look like?
The aromatic ring with an OH.
Nope. I mean, if it had an OH, it would be an STY, right?
Mm-hmm.
But it's not an STY.
I don't know. Hold on.
So how about the ones that are charged? Which amino acids would be charged?
Ones that are acidic and basic.
All right. What do those include?
Tamic acid, aspartic acid, glutamine, aspartate.
No. The first
Oh, wait. Um, okay, so the two acids and then histidine, lysine, H, K, arginine.
so these so this is aspartic acid and the D. and the E is glutamic acid, and these would give you negative charges because they're carboxylic acids and they donate protons. And then the basic ones are H histidine, K lysine, R arginine, which are, since they're bases, they get protonated, so they're positively charged.
Mm-hmm.
So here, if you don't remember, each of these, you could at least know that they're not charged because they don't contain any of these, right?
Yes. So
So
it's 0.
yeah. Now,
But let's go over the amino acids because I'm still really struggling with this. Okay.
so STY must include hydroxyl groups.
Right. So serine, tyrosine, and phenyl
So
and tryptophan.
phenylalanine is gonna start with an F.
Right.
That is gonna just
Why? It's because tyrosine is the only one that has a Y in it.
uh-huh. I mean, technically tryptophan has one too, but yeah, the phenylalanine is just gonna have a phenyl group, which is a benzene group. Tyrosine Y is gonna have the benzene group with an OH. So just remember STY, they all have OHs, right?
But so this isn't tyrosine?
No, the Y is the tyrosine.
Is okay, so then T is
No,
tryptophan.
threonine, yeah.
What is tryptophan?
Tryptophan has a
W.
yeah for W, and it's a double ring. It's like something like this.
They're like confusingly similar.
Well, just know that phenylalanine over here, the phenyl part means benzene. So phenylalanine must have a benzene. Tyrosine, if it's part of STY, it must have a hydroxyl group. So it's a phenyl group attached to an OH group. That's tyrosine. Tryptophan has 2 rings, and it's the only one. Well, it's the only amino acid with 2 rings, so that can help. And you can then have the um the rings that have nitrogens in them. So like proline can have that, and histidine can have that.
Okay.
But ultimately, STY has hydroxyls, and if you're mixing up like T with tyrosine, you can kind of remember that tyrosine has the Y in it and could be the Y. And I know tryptophan has a Y in it too, but tryptophan is not going to have hydroxyl groups, so tryptophan is not going to be included in STY. So,
Okay.
yep.
Researchers use which type?
Oh, you probably chose beta because you looked at that.
Yeah.
Tell me why that won't be it. Like, what's that beta? Talk
It's
about.
the amount of radioactivity was detected using beta decay.
Yeah.
Beta decay is in beta ATP. It's different
Oh,
things.
yeah. Beta decay is a type of radioactive decay, right?
Yes, and we're talking about ATP, which is mentioned.
So remember when we talked about, for instance, like STY taking a phosphate from ATP, and ATP has 3 phosphates. How were we able to distinguish which like phosphate is which? What we used like a naming convention.
I don't remember this. What?
So ATP has 3 phosphates, and how do we distinguish those three phosphates?
Alpha, beta, gamma.
Yep.
So Y is out. Or no, that is gamma. This is rho.
That's lowercase delta.
Whatever, still wrong.
Mhm.
Um, uh,
So we're talking about super high yield topic of kinases here, right?
yes.
And kinases use STY to remove one of the phosphates from ATP, right?
Like ADP.
Uh-huh. So which phosphate does it remove?
Isn't it? It probably starts from gamma, right? The ones that are further away.
Uh-huh.
So if it's removing this, it's C.
Yep, that's the gamma one, right?
Okay.
Like
So we would always start with is it the smallest? Like what is like, I
it,
can't remember what the alpha, beta, gamma stand for as far as it's just labeling them.
if you move outwards, so adenine, first phosphate, second phosphate, third phosphate,
Oh, this is the gamma, this is the alpha.
this is gamma, yeah.
Okay. All right. Makes sense.
Yeah. But I want you to see that if you go back real quick, that this is what they want you to do. They want you to look back,
C
see,
beta. And
and choose beta. And that's what you don't want to do. You want to make sure you understand what they're asking, and you want to make sure you understand what they're saying in the passage instead of the mix and match game. So
Mhm.
be careful there.
I think for this one, I did the same thing because I saw serine and threonine.
Yeah, but that still wouldn't make sense because those are not charged.
Oh, right. Okay.
So, and also that passage was about kinases, and I keep saying kinases are super, super, super high yield, and you're
And
seeing
kind
that a lot.
of
So you got
uh,
to know
use
about that.
ATP and phosphokinase use phosphates.
Wait, what's up?
Like there was something like kinase uses ATP, but then phosphokinases like don't use oxygen. So they're like
Uh,
no
Kinases
ATP.
are just going to be transferring phosphate, so that could either be taking the gamma phosphate from ATP and then transferring it onto the substrate to phosphorylate the substrate, or it could mean taking the phosphate from the substrate and giving it to ADP to make ATP. It can do both of those.
Okay.
Yep.
All right. This one, it's
Mhm.
well, I know it's not hydrolysis because it's
Mhm.
dehydrogenase,
Mhm.
and then it's O to OH. Is it carboxylation? Like carboxylic acid?
Um, what does the carboxylic acid look like?
Oh, there has to be an oxygen. Never mind.
So there's three ways you can do this.
Like I was looking at these and it's obviously not getting reduced or oxidized. on this side, but then there's the only change happening is between the O and the OH.
Yep. Mm-hmm.
We see that there's lactate.
Yeah, does that enzyme like what enzyme class is that?
It's getting oxidized.
How do you know?
Is hydrogen to a positive charge.
Uh-huh. Yeah. So yeah, there's three ways we can answer this. One way would be, okay, if we're using a dehydrogenase, if we knew that that meant oxido-reductase, we would know that we're doing some redox reaction, right?
Yeah.
That's one way. If you didn't know that, we can see what happens to this NADH. All right.
Mm-hmm.
You can see what's happening to the NADH here.
Oxidized.
Yep. So NADH gets oxidized into NAD plus. That's another way. If we didn't see that, we can also see, okay, if we're going from a carbonyl to an alcohol, right? We're doing something there too, right? Right?
Yeah.
So remember that question that said,
Yeah. It's
what's up?
Isn't it going from a tertiary alcohol to a secondary alcohol?
Where do you see an alcohol in compound two?
I don't know. Never mind. Forget I said it.
So remember that question that was about lactases? And they said that lactases oxidize hydroxyl groups. So we had like
Yeah.
this turn into this, right?
Yes.
And that's if you oxidize it, right?
Mm-hmm.
What we see happen is what?
The opposite.
Yep.
So
So,
reduce.
yep. So if you knew this gets reduced to give you this, that could be your third way of figuring this out. So I want to make sure that you have like multiple ways to figure out an answer. So these are the three different ways. So one could be if you knew that dehydrogenase is an oxidoreductase, and that's something you should know because you should be able to see an enzyme and know what enzyme class it falls under. So dehydrogenase means it's an oxidoreductase, which means redox happens. So we can get rid of that right away. If we didn't know that, we can also see that NADH gets oxidized as it turns into NAD plus, which means that the substrate itself will have to get reduced. So that can help us figure out that it's B. But if we didn't know that, we can also see that, hey, we're taking this carbonyl group, right? Or ketone group, right? And we're turning it into just a hydroxyl, just an alcohol there. So we're losing bonds to oxygen, which means that we're uh, reducing it. So it's just the reverse of this up here. So those are the three ways to figure out the answer for this question.
Okay. All right. What time are we going till today?
Until like 5:30.
Okay. Okay, so I want to ask you what these two things are because I've heard of Doppler, but I don't remember like Like I know I've heard it. I just can't remember what it applies to, and I've never heard of this one.
Got it. Yep. So the Doppler effect involves
Hearing or something?
so essentially, let's say we have some car moving in this direction and it's giving off sound. Let's say like these are the sound waves of a certain frequency. And if this is you, and either the source of the sound is coming closer to you, or you're coming closer to the source of the sound. Either way, if you're getting closer to the source of the sound, you will observe a higher frequency. So if you imagine like an ambulance, if it's coming towards you or if you're going towards it, the pitch of that sound goes up. And pitch is just a way of expressing frequency and sound. Until it's like right there where you are,
And
at
then it's
at which the frequency that you hear is equal to the like the actual frequency of the siren or whatever. But then once it passes you, since the distance between you and that source of that sound is now increasing, you will observe a lower frequency of that sound, a lower pitch. So, and this can happen with light as well. So one of the things that we can do is we can observe some planet or something that's really, really far away, using like, really powerful telescopes. And let's say the planet is supposed to be like orange or something, but it appears more like a reddish orange to us. It means that, well, the wavelength of red is higher than the wavelength of orange. So it means that it's moving away from you. And that's one of the ways that we know that the universe is expanding, because we see what's called a redshift, which is essentially the same thing as the Doppler effect for sound, except going from orange to red, you're increasing that wavelength and you're decreasing that frequency. So it's just like the same thing as something moving away from you and the frequency going down and the wavelength going up. So that's Doppler. So Doppler involves any type of wave, meaning including sound and light, and your observed frequency goes up if the distance between you and the source is getting smaller, and that frequency that you observe goes down if the distance between you and the source of the sound is getting larger.
Okay.
So that's Doppler. Now, Venturi effect is something like this. So let's say that I'll kind of draw this thing. This, and oxygen is moving like this. Now, let's think about it in extreme terms. Like, let's imagine the oxygen moving in this direction is so, so fast that it sucks in or creates, like, where these openings are. It creates like a vacuum on the inside here, which causes air from the outside to get sucked in through these holes. So if you imagine that, I used to do like kind of like a demonstration with like a straw wrapper, where I would like kind of like bend it like this and put my fingers on either side to kind of anchor it, and I blow like air like underneath, underneath it, do you think the height of this part of the straw wrapper would go up or down?
Up.
So it'll actually go down because if you're blowing underneath it, the air underneath it gets to be smaller because it's moving past it. So the air underneath it, there's less of it, and that creates a low pressure in like environment, which will cause the top of the straw wrapper to get sucked downwards. So that's kind of like what we're doing here. If the oxygen is going in this direction, imagine it's going so fast that the air molecules, or sorry, the oxygen molecules here are no longer there. They're, let's just say, over here, which means that there's a vacuum here and here, which means the air would be sucked in at those regions. So
Okay.
basically, it's an inverse proportion between velocity of the air, or in this case, the oxygen, and the pressure. It's kind of like how a wing works. Like the wing will have a high velocity above compared to below. So high velocity but low pressure, and underneath it's low velocity, high pressure. And that means that underneath the wing is a higher pressure than above the wing, and that causes lift. It causes like the overall wing to go up. So so basically just think of it like that. If it's a high velocity, like this oxygen going through this mask and into the person's mouth or whatever is going fast, and the faster it goes, the lower pressure this region is. And if it's a lower pressure region, it will suck things in from the outside. So that's also why in like a hurricane, windows don't blow in words, but they blow outwards, like they get sucked out.
Okay.
Yeah.
I feel like I don't understand how this relates to like this problem. Like I get that the air would go down. I get this part,
Mm-hmm.
but I don't know, like
Mm-hmm.
this was about Venturi.
Uh-huh. Yeah, yeah. So
Okay.
like essentially, if you think about it like this, so you have some tube with openings, right? And moving through the tube is, let's say, oxygen. And if you like kind of suck inwards like the oxygen, we're moving all this like stuff to the right. And if we're moving it fast, it means that, like, let's say this is a molecule of oxygen, right? It's moving this way. So we end up having not many molecules of oxygen in this region since it's being moved. So what we then have is a smaller amount of oxygen near the gaps, which means that there's going to be if we have a low amount of oxygen there, we have a low pressure region there, which compared to the outside, which is a higher pressure, will cause the air to get sucked in through those holes.
Okay. All right.
Mm-hmm.
Causes. Okay. So remember.
Imagine taking a water bottle, make a bunch of holes in it, and let's say you put it, submerge it in water, and you suck inwards, right? Initially, the water bottle doesn't have any water in it, but it has the holes. But let's say you submerge it in water and you suck inwards from like the opening.
You would get water.
Yeah, water will get sucked in, right? So I know that was using water as an
Oh,
example.
but I get it. This is like a good example because I can visualize it. Okay.
Yeah. Like
Right.
another one could be like, let's just say you have a colored smoke. You have green smoke or pink smoke. If you suck inwards, the pink smoke or whatever will enter into the bottle. So,
Okay.
yeah.
All right. With this one, I remember like cut pyramid.
Mm-hmm.
So I knew it was van der
Oh,
Waals.
yeah. Oh, so this is yeah, so I guess this is like a content thing that we just have to know. So basically, I mean, we know that greater CG ratio means higher
Right.
temperature because
Yes.
CG can make 3 hydrogen bonds as opposed to 2 from AT. But this is some other stuff that they're testing us. It's something called pi stacking. And that's when you stack nucleotides together and you can get overlap of uh the bases. So essentially it's going to look like this. Wait one sec. It'll look like kind of like this. Oops. Here in the chat, put an image. So yeah, you could see like AT, like A here, T here, G here, C here. And yeah, it's what we expect. For instance, 2 hydrogen bonds between A and T, 3 hydrogen bonds between C and G. But in addition to that, you can see how the rings of the bases could be stacked on like atop one another to create this type of bond, which is more vertical, and that's called pi stacking.
Okay.
Yeah, and that also enhances the stability of that of the DNA molecule.
All right.
Yep.
Okay.
Mhm.
Mhm.
That's another redox.
Okay. But I was thinking of it like overall, because remember we were looking at like net and it's like half reactions.
Yep.
So I knew it was something like that where it's like this to this. Wait, but like that's what was confusing to me is that they don't relate.
Yeah, so it seems like reaction 1 is the actual overall reaction, and then the 2 half reactions are the half reactions that, you know, produce that overall reaction. So we could just look at reaction 1 here.
Okay.
So why don't you assign oxidation states there?
This gets reduced.
So what are you assigning? What oxidation states are you assigning?
I think that nitrogen gets oxidized and hydrogen gets reduced.
Yeah, but I'm asking what oxidation states do we have? To figure that out, we need to figure out the oxidation states, right? 'Cause otherwise, you're just guessing.
Well, these are both neutral. Like neither of them have a charge, but you can see that it goes from N2 to N and H2 to H3.
What's the oxidation state of N2?
What oxidations means?
Oxidation state? Like, you know what that means. Like, that's the... So, H2O, the hydrogen has an oxidation state of something. What would that be?
Oxidation state?
Yeah.
I literally have no idea.
So +1, and we have two of them, so it's +2.
Oh, and then oxygen is -1.
Nope. If it was -1, H2O would have a +1 charge.
And so it's -2.
So oxidation state is the like the number of valence electrons that an atom in some molecule would have. And you need that if you are going to figure out if it's redox, because that's what redox does. If something gets oxidized, it gains electrons. Sorry, loses electrons. If something gets reduced, it gains electrons. So you can't answer a question like this if you don't know any oxidation states.
Okay.
Because how else? Yeah. So N2 gas, oxidation state of 0, H2 gas, oxidation state of 0. Right?
Mm-hmm.
And now what would it be for the nitrogen in ammonia and the hydrogen in ammonia?
Okay, so nitrogen has a -2 and hydrogen is +2.
Sorry. So
NH
why would hydrogen have... So what's the oxidation state for one hydrogen molecule?
for one hydrogen, it's +1.
And we have how many here?
It's 3. So which means that nitrogen has to be -3. Why are these two 0?
Because N2 gas and H2 gas are just, they're diatomic uncharged particles, so they're not going to have any oxidation states. Because I mean, you have, like two H's, right? Like, one could be like -1, one can be +1 to give you net oxidation state of 0. So for these diatomic elements, their oxidation states are going to be 0.
Okay.
Right?
Mm-hmm.
So what happens?
Well, then based off of this one's at the anode and this one's at the cathode.
How are you making that choice?
Because negative charge,
Because of what?
like negative, positive.
What does that have to do with it?
I don't know.
So what are we looking for?
The location.
I mean, what are we like?
Figuring out which one's getting reduced and which one's getting oxidized.
Yeah. So do that.
This one's getting oxidized and this one's getting reduced.
What happens when something gets oxidized?
Gains a positive. No, gains a negative.
So no. So don't you remember like Leo, Gur? Never saw that.
This is electrons. Gains electrons. So reduction, it gains a negative charge, and oxidation, it gains a positive charge.
Is that what this says? What does this stand for?
Oxidation loses electrons. Reduction gains electrons.
All right.
Means that when something's being reduced, it gains a positive charge. Oh, it gains a negative charge.
Yeah.
Was I saying the opposite? Or
Yeah.
all right.
Yeah. So now what happens?
Well, then we can figure out that like which one is reduced and which one's oxidized, but I don't understand how that.
So just do that.
Okay. Nitrogen's getting reduced.
Yep.
Hydrogen's getting oxidized.
Yep.
So A and D are out. Wouldn't the anode have a negative charge?
That doesn't have anything to do with this stuff here. So another thing, I guess, that I learned in like middle school was like anox, red cat. So for this, think of Leo the lion says Gur. For this, right? Right.
Mm-hmm.
Leo the lion says Gur. Right? What are you thinking of?
Oh, well, if anode is oxidation, then
So this over here. Just so you could remember it by thinking Leo the lion says Gur.
right, and anox red cat.
Yeah. So this shows you what happens with oxidation and reduction. And then this shows you where things will be oxidized and where things will be reduced. You definitely have to know this. This is like very like elementary. So yeah, I added it to the list of things for you to go over.
Can you text me this list, please?
Yeah, I'll do that. But so okay, so now that we know where oxidation takes place, where reduction takes place. What can we do?
Well, can't you just apply it if oxidation is at the anode and redox is at the cathode, then we know that nitrogen is cathode, hydrogen is anode.
Yeah. Cool.
This is another bond one.
Yeah, so this is when you have some transition metal that's surrounded by a bunch of things like H2O or NH3, and the overall charge still is not 0, it's like 2+. It's called a coordinate covalent bond. So this is just a content thing.
So coordinate covalent is for transition. What is coordinate ionic?
Oh, that's not anything.
Okay, so this doesn't exist.
Mhm.
Can I ask you a stupid question? Can we
Ionic bond is going to be a bond typically between a metal and a non-metal, and it's going to have, there's going to be a large difference in electronegativity between the metal and the non-metal. So ionic bond could be something like NaCl, things that typically can dissociate in water, for instance, but will involve bonds between a metal and a non-metal. So, and we know that metals are going to have very low electronegativity values, and the chloride and, you know, the halogens and things like that, the non-metals will have higher electronegativity. So you're going to have a huge difference in electronegativity between the two, and it's going to be forming a, you know, like a dipole. Covalent bonds don't include metals, they're just going to include non-metals, and that's typically what we see for like organic chem, where pretty much it's hydrocarbons. We're not dealing with metals there. So that would be like CH4, you know, CHO, like all that stuff. Pretty much, you know, if it's bonds between non-metals, it's covalent. Yeah.
Okay, so two nonmetals covalent, metal and nonmetal is ionic. Coordinate covalent is
Also metal and non-metal, but it will involve several non-metal. Uh, or maybe I should put it like this. The non-metal is for coordinate covalent will be an actual molecule rather than a single atom. Because in ionic bond, you have a single, you know, metal atom and a single like non-metal atom. But if you had something like H2O or NH3 as your non-metal, it's not just a single atom, it's like an actual molecule. So that's what you'll see for coordinate covalent. And also ionic ultimately produces something that has a neutral charge, but a coordinate covalent bond will still have that cationic charge.
okay.
Hmm.
All right.
Yep.
All right.
So I think we can, yeah, wrap up here. So I would say to do your blind review for this test
Mhm.
and, you know, followed by your regular review. And
When are we meeting again? Are we
yeah,
doing tomorrow?
so
Wednesday?
let's see here. So yeah, I have to hear back from,
Because we scheduled Thursday at 6.
yeah, so yeah, we're definitely gonna see each other then. But I'm gonna see if I can move people around for Tuesday and Wednesday. If I can't, we might have to meet late, like 8 p.m.
I'm fine with that. I just, as long as you, I'm sorry. I like really should have scheduled. I don't know how it.
So So yeah, tomorrow let's meet at 8, and if anything, changes and I have like another opening or something, I'll put it in for earlier, but as of now, let's meet at 8 p.m. tomorrow. Works?
Okay. Yeah, perfect.
Awesome. All right, congrats on taking that test, and yeah, I will see you tomorrow, okay? Take care.
Bye.